All right. Good morning, everybody. Um, Before we start with the presentations, I want to talk a little bit about the final exam, and also in that process, give people a little bit of advice um, on when they have some concerns and how perhaps best to address those. Um, so scheduling an exam that spans multiple universities that have different calendars is always going to be logistically a bit challenging for us, especially since the deadline for us reporting grades varies by institution, and particularly if people are graduating this spring, it means they have to have the grades even sooner. Um, these exams are a little bit challenging on our end in terms of the fact that once the exams are turned in, we then give each part of it to the respective instructor to grade their portion of it. We then need to compile them all and come up with a composite score, and then we have to evaluate the overall score for all components of the course over the course of the semester, and submit final grades. And some of those deadlines for us are basically by the end of next week. So we have a pretty tight time window for us to actually complete this whole process. We received a few emails from folks, some of which were done very, very nicely, asking about being able to have an extension on the deadline that we gave 3 weeks ago on when the timing of the exam was going to be. But some of them were not quite as well put. I'll say that. So I think it's important when you're communicating with others about your situation that you try to put yourself in the position of others, and be careful about making statements that imply that others may not be considering your particular situation very much. I know many of you have jobs like many of us had when we were in school as well. But that is part of the reason why we give over 3 days, close to 4 days for exams. And we try to always span them so that way they have multiple days that are during the week as well as in the weekend, because we recognize that people have different time constraints that need to be considered. So we do consider what is important to students. And also you need to recognize that these exams are completely open note. You don't have to do studying for them ahead of time, like you do an exam that then takes you 3 hours afterwards. And if you work your way through these with your notes, these are exams that really should only take a few hours. So we think that the time frame that we allot for these exams is fair. We had to figure out exactly what our reporting deadlines were, so maybe we gave you a little less notice than we would like to in most years, but we did state exactly what they were going to be 3 weeks ago. So with that said, a few of you did write some well put statements explaining what your situation is. We have a very narrow window to complete our grading of these exams, but I am willing to accept exams as late as noon on Monday. That is now going to be a complete 4 days to complete an exam that should really only take a few hours. So I know people are busy and they have a lot of their commitments, but that you should be able to have time to complete it during that window. And I just, again, will advise you that when you want to complain about a situation, it is best to consider that, other people also have considerations that need to take into account, and it's important that you recognize that, and that there are a lot of your other classmates that are going through very similar situations that you are, and we are aware of what those different obligations we all have, because many of us had to do work jobs and other things while we were in school as well. So again, we're going to allow the exams to be submitted as late as noon on Monday. It's really the latest we can do that while still giving our instructors enough time to complete their grading of the exams and for us to get the final grades in. And so that's all I'm going to say about that. Hopefully that is something you guys can then work through in terms of managing your own time to complete the exam. So with that, we're going to move on today, and we're going to have the first of two sessions of the class that are going to be about presentations that the graduate students are going to give on a topic of their choosing that had some suggestions that were given to them about topic areas that might be appropriate for this course. These, I'll remind you, these work just like all of the other lectures throughout the semester. So these are part of your exam. So you should be paying good attention to this. We are making all of the presentations available like we do throughout the whole semester. And there will be so there will be questions associated with each one of these presentations. Additionally, the graduate students have part of their grade is the presentation and the discussion. You should feel free to, just like you can throughout the semester with the lectures, to ask questions. We want that. We want these to be discussions and to be interactive, so we encourage you to do that. And the way it's going to work, a couple of the students have expressed a little bit of concern with being the presenter and running their PowerPoint presentation through their internet connection. So what we're going to do instead is Mert is going to actually pull up the PowerPoint, and she will wait for cues from the presenter to say next slide, and then she'll advance the slides. If they want something highlighted, I suggest that you indicate, you're talking about something in the upper right-hand corner of the slide, and then Mert can indicate where that is located. Then we'll have a little bit of transition between each one. We're going to have 4 presentations today and 4 on Thursday. And I think that's all I wanted to say about that. I see in the chat people asked when the exam is going to be posted. As I said earlier in the class a couple of weeks ago, that the exam will be posted in the afternoon on Thursday. We have to have time to actually write those final couple of questions about Thursday's presentations. So it won't be immediately after class, but it will be during the afternoon on Thursday that we'll post the exams. Any questions about how this is going to work? And yes, the question about grad presentations will be on the final, as I just said. So this is just like our other lectures. It's just today and Thursday, they're given by 4 different people.
Okay, yeah. I just got on here because my laptop wouldn't let me log on, so I had to call by my phone. What did you just say about the final?
The exam will be posted on Thursday afternoon. It will include these graduate student presentations. And remember, it also covers the field trip. And we are going to allow people to turn in exams up until noon on Monday.
And it's this Thursday?
This Thursday.
Okay.
Any other questions? Okay, Mert, I'll turn it back over to you, and then you can pull up the slides and we can go from there. Okay?
Sounds good.
All right, thank you.
So Caitlin, you're our first.
Yes, can you hear me?
I can.
Okay, perfect. Do I need to turn my camera on?
I don't think so.
Okay. Okay. All right. Well, I'm Caitlin. I am a student at LSUS in Shreveport, and my topic is coastal change and human resiliency. Next. So the dictionary defines resilience as the ability of a substance or an object to spring back into shape. In this instance, obviously, we're talking about how people are resilient to the changing coast. And then I'm going to correlate it a lot with how they use adaptation in their resilience. And adaptation is a change or a process of change by which an organism or a species becomes better suited to its environment. All right, next. So Thomas Wilbanks described 4 key elements of resilient communities. The first is the ability to anticipate disruptive events, the capability to respond to them effectively, the mechanisms to recover from them equitably and efficiently, and take steps to reduce vulnerabilities to future events. So throughout my presentation, I won't really revisit these specifically, but you will see kind of examples of how humans use kind of these 4 key elements to be a resilient community. Next. All right, so just some key points I'm going to make. The first is obviously how the coast is changing. 2 that I'm really going to look deeper at is coastal erosion and temperature. It's 2 that we've talked about a lot during this class, but I'm going to revisit them. How humans are adapting to these changes, and I'm using pretty much just Louisiana in my presentation. Even though a lot of these things are being used pretty much everywhere, not just Louisiana, and actually not just in the United States. Resilience after hurricanes, specifically Hurricane Katrina, of course, and resilience after oil spills. Next. All right, so first thing, coastal erosion. So coastal erosion is turning marshes into open water. So the marshes and the coast pretty much is disappearing, and everything is just turning into open ocean, which we saw when we went down there. Really, we saw how a lot of the marsh had disappeared, and we've talked about it a lot during other lectures. Some things that the loss of the marsh really leads to is waterlogged soils and iron toxicity, physical and biological structure of fish and shellfish habitats are disturbed, which in turn can disturb the prey-predator relationship. It can disturb, when fishermen go out there, that's just kind of their livelihood. It can disturb that. Next, larval stage of some fish feed on copepods in marsh waters and can't survive in the open water. And then the frequency, duration, and strength of flooding affects the accessibility of the organisms that live in that area, and also of, the humans. All right, next. Ultraviolet light. You'll see in my little diagram how it's different. It penetrates different in open ocean compared to the coastal water. So it goes way deeper in the open ocean than it does in the coastal water. Some species that live out there can't really, they need certain ultraviolet light to really survive and to, live just the best way they can. And so it really messes up how they live. So with greater expanse of open water, estuarine surface waters will be exposed to greater levels of ultraviolet light. which affects the food supply for larval fish. Copepod eggs are usually released at or near the surface. And then, so the hatching success is severely reduced by exposure to natural levels of ultraviolet lights. And then that would be the levels that the eggs would usually be exposed to in open water near the surface water. So when they are, what the light they would usually be exposed to in open water near the surface changes. Now they would be exposed to it coastal water near the surface. Next. So coastal erosion also affects temperature in these areas. As you'll see on the right side, my little shrimp picture, a rise in 4 degrees Celsius in summer water temperature in shallow estuaries would exceed the lethal limit for white shrimp when they are at their peak larval stage, post-larval stage. So this could mess up the whole food chain. It could mess up, you know, how shrimpers get their shrimp. It could mess up, you know, a lot. And then also on the other side, some other changes that altered temperature could cause: advanced estuarine spawning cycles of fish. Which means it would make them speed up. It could also slow down the spawning cycles of the fish, which changes the synchrony of yearly life stages with their supply of much needed planktonic food. It would lead to distribution shifts. So in areas where you would once find temperate and subtropical fish, you would find subtropical and tropical fish. Increased health risk from, this was, the next two are for the human species. So it could cause increased health risk from storms, flooding, and waterborne illnesses, droughts, heat waves, and declining air quality. The heat, rain, and humidity help spread infectious diseases as pathogens come in closer contact with humans. So it could really affect the health of humans. Also, the health of a lot of those species that live in those areas. Next. So, how are people coping and adapting to this changing coastline? So the health department does have an environmental tracking program collecting and distributing public health data to guide policies. So they're just keeping track of kind of the I guess the changes in the weather, I guess, so like the humidity, the storms, and that helps them keep better track of what the human populations are going to be more susceptible to in these areas. So that might change as the weather changes and as the coastline changes. It will change what people in the area might catch or what they might be exposed to health-wise. So they do keep kind of track of that. Utility companies are committing to stabilize greenhouse gas emissions. Energy Corp cut their carbon dioxide emissions by 69 million tons over the last decade, which is a lot. Increased irrigation during extreme hot or dry months to maintain crops. So all these are examples of really how we're adapting and we're changing how we respond to these changes in the weather and these changes in the coast. So this one we've been over. They're building houses and businesses higher off the ground because of increased flooding risk. They're also publishing more guides like Adapting to Coastal Climate Change, which is kind of a guidebook for development planners. And there was one called Adapting to Climate Change and Rising Sea Levels. So this kind of just gets it out there and helps other people learn to adapt to these changes also. And then the Green Army was formed. And this is an alliance of groups and individuals that are fighting for clean air, clean water, and healthy communities in Louisiana. Next. This is just some pictures of, these are the 2 books I mentioned, and then, you know, a house on stilts, because you haven't seen that yet. Next. All right, another cool thing I kind of wanted to talk about was carbon sequestration. Sorry, I'm not really good at saying that. So this kind of just goes with not as much as the changing coast, but as a whole global warming, which is causing changing coastline. So it kind of all correlates. But I did just want to talk about this real quick. So carbon sequestration is both natural and deliberate processes by which carbon dioxide is either removed from the atmosphere or diverted from emission sources and stored in the ocean, terrestrial environments, and geologic formations. So there are 3 types. The first, organic carbons. The world's oceans are primarily long-term sink for human-caused CO2 emissions. It currently accounts for a global net uptake of about 2 gigatons of carbon annually. This uptake is not a result of deliberate sequestration, but occurs naturally. So this occurs just naturally in the atmosphere through chemical reactions between seawater and CO2 in the atmosphere. Next is the terrestrial carbon sequestration. It's typically accomplished through forest and soil conservation practices that enhance the storage of carbon, such as restoring and establishing new forests, wetlands, and grasslands, or reduce CO2 emissions, such as reducing agricultural tillage and suppressing wildfires. And then the last one is geologic. This begins with capturing carbon dioxide from the exhaust of fossil fuel power plants and other major sources. And then it's then piped below the land and injected into rock formations. Next. So these are just 2 illustrations. The first one is the biological. So this one, you'll see CO2, it goes through trees, down into the soil, and then back out. And then the second one to the right, you'll see the terrestrial, and then the, what was my other one? Geologic. So you can see how it's pumped down into the ground from the big buildings over here, whatever they're called. They pump it down into the ground and they store it in this rock down here. So those are your 3 types. All right, next. Some more adaptations that humans are using to be resilient against the changing coast. They're water reuse and recycling, protecting wetlands to reduce damage to water quality from severe storms, increasing green space and decreasing paved surfaces in urban areas to reduce runoff, slow the rate of water table depletion, and reduce the impact of extreme precipitation events. Increase the use of air conditioning is associated with an increased demand in electricity, which can impact water availability and regional water ecology. Next. All right, so now I'll talk about Hurricane Katrina. So it hit as a Category 3. It caused 53 levee breaches, $125 billion worth of damage, and then to the area it caused beach erosion, massive tree loss, and several oil spills. Next. Oh, this is kind of out of place, but I think we've seen this in a different PowerPoint anyway, but that's just tells you how much land loss we've seen in the past. And what is you're projected to see. Sorry, it's out of place. Next. Okay, back to Hurricane Katrina. So how humans have adapted and kind of tried to protect ourselves from future damages as huge as Hurricane Katrina caused. So more than 36 miles of barrier shoreline has been restored. And then the barrier shoreline should be able to provide a better kind of barrier between the effects of a hurricane and the coast. So the basin is better positioned to provide coastal resilience in the face of future storms and sea level rise thanks to these efforts. Next. So last, I'll talk about the oil spill real quick. So the Deepwater Horizon exploded, dumping over 200 million gallons into the Gulf. It caused a large number of dead dolphins and heavily oiled places, and not only dolphins. Well, it'll tell down here. Evidence of 1,200 square mile area of oil contamination on the ocean floor. Studies estimate 800,000 birds died as a result, and between 20,000 and 60,000 Kemp's Ridley sea turtles died as a result of the spill. A 2014 study found concentrations of PAH, which can cause harmful effects in many birds, fish, and wildlife in the marshes, which may persist for decades to come. Next. So some things that we have done. The Restore Act was established to provide funding to the Gulf region to restore ecosystems and rebuild local economies. The MCDA is used, so that's an approach, it's used to just analyze situations and help make decisions about situations that might be harder to make because of it might be harder to solve analytically. So it's the MCDA. It's an approach especially used for examining sustainable alternatives in oil spill management crisis because it provides a process for integrating economic, environmental, and social impacts, which are not easily included in traditional analysis methods. And also, Healthy Gulf, Healthy Communities Project sought to conduct research to answer questions for the affected communities. Next. Oh, that's it. I'm done. Does anyone have any questions I may be able to answer? I don't know. If not, that is it for me.
All right. Well, thank you. Sure nobody has any questions?
What was the program that you last said?
Sorry, I muted myself. The multiple criteria decision analysis, it's just a program they use to help make decisions that are kind of hard to make when you can't do them like analytically. I didn't really look much into it, but there's a lot of stuff about it if you wanted to look it up.
Okay.
This is Guillaume Riuco. Just a follow-up on the question. Is it a statistical analysis, a way to predict, to develop prediction? Did you read about it?
Oh, are you asking me? Yeah,
Yeah.
it helps. Well, it really helps. So it helps kind of examine what happened, yeah, and predict give like similar scenarios that could also happen, yeah. Like predict future things and how you would respond to those situations.
Okay. And do you have an idea of the kind of parameters that people are putting in this model?
Oh no, I don't. I didn't look that far into it.
Okay. As you mentioned it during your presentation, I thought you dig more about it. Thank you.
Okay, thank you very much. I think we should probably move on to make sure we can get the other 3 in today. Mert, can you load the next one up?
Hi, can everyone hear me?
Yes.
Okay. Hi, everybody. My name is Juita, and I attend University of Louisiana at Lafayette. And today I'm going to expand on the topic of PPCPs and its a few effects on the coastal ocean. Next. I will briefly be talking about PPCP sources as a recap from Dr. Roberts' lecture, why coastal environments are at risk, a few types of PPCPs, as well as their effects and potential ways to mitigate this emerging issue. Next. So PPCPs stand for pharmaceuticals and personal care products. They include prescription and over-the-counter drugs, as well as veterinary medicine, sunscreen, bug repellent, things of that nature. This flow chart demonstrates from the top left different ways that PPCPs can enter the environment and affect different trophic levels. The purple dots depict the different organisms that have accumulated PPCPs in their tissues. Next. So can you click on the mouse for the photo? The photo should pop up. Sorry, thank you. So a few sources of PPCPs. The main one is through wastewater treatment plants. And wastewater treatment plants have a varying range in which they're going to treat their wastewater, thus resulting in various discharge rates of PPCPs into the environment. Can you click again? Another source would be veterinary care for livestock, as well as aquaculture, as you can see in this photo. There was a review by Gaw et al., and they found that 75% of medicine used in aquaculture dietary procedures can be lost to the surrounding environment. Can you click again? Oh, can you click again? Sorry. Landfills and sea fills also deposit PPCPs into the marine ecosystem through liquid runoff, as well as ships are allowed to discharge treated and untreated sewage at specific nautical miles away from the land, causing direct influx of these PPCPs straight into the marine ecosystem. This photo just shows the different landfills that are pretty close to coastal lands or coastal areas. Next. So coastal and marine environments are extremely susceptible to the negative impacts of PPCPs due to the high population sizes observed in these coastal zones. 21 of the 33 megacities are on the coast, and megacities are defined as cities with 8 million or more inhabitants. With an increase in population size, we can also see that there will be an increase in urbanization, And with an estimate of 49% of marine ecosystems are negatively affected by anthropogenic stressors, such as eutrophication and overfishing, now we have to add PPCPs onto that list. Can you click again? According to the Department of Environmental Science of New Hampshire, the presence of PPCPs have been shown to impact aquatic species, such as a change in sex ratio in fish populations, development of female characteristics in male fishes, and unfavorable effects on invertebrates. Next. In 2004, a review by Gall et al. Found over 100 pharmaceuticals and their metabolites detected in coastal waters. Most of these compounds that were detected were antibiotics, but just to mention a few, painkillers such as acetaminophen and ibuprofen were found, antibiotics such as sulfamethoxazole as well as triclosan were also found, and antihypertensives. Next. So what are some of these PPCPs? I'm just going to cover two. Triclosan was first registered as a pesticide in 1969, but it is currently found in many consumer products, and it is used to slow or prevent bacterial, fungi, and mildew growth by inhibiting fatty acid synthesis in these organisms. It can be found in things such as toothpaste, deodorant, and hand soap. Sulfamethoxazole is an antibiotic commonly used to treat illnesses in humans, dogs, horses, cattle, pig, etc. And it does this by inhibiting folic acid synthesis. Next. Okay. A pilot study was conducted in 2009. It was the first of its kind to look at a wide range of accumulation of PPCPs in fish tissues across the United States. Here's the map of their study sites, including the one with the star is their control site. And they chose these 5 sites for the reason that they had high effluents from wastewater treatment plants. And their control site was fairly pristine. They tested for 24 different pharmaceuticals, and 17 were not detected. And they also tested for 12 personal care products, but 10 were not detected. Triclosan was detected, but only in acute amounts. They found that sites that had, they found that sites with more advanced wastewater treatments tended to have less concentrations of the PPCPs, and sites with less advanced treatment had more, which is what they expected to see. They also found that these pharmaceuticals and personal care products were observed in large concentrations and more frequently in the liver tissue than in the fillet tissue. At least 2 of these compounds were detected in their liver samples from all 5 sites versus only 3 from the fillets. This suggests that the accumulation of PPCPs in tissue has a relationship with the amount of wastewater treatments that is given before they are discharged into the environment, as well as the demographics. Next. This photo shows the results from the Chicago site with the most PPCPs found in the fish were an antidepressant, followed by an anti-seizure medication and antihistamines. Sorry for the really small font. Next. In order to showcase another effect of PPCPs, I'm just going to quickly talk about Vibrio. Vibrio is a genus that is naturally occurring in marine ecosystems. They are a genus of pathogenic bacteria. In the United States, Vibrio vulnificus and Vibrio parahemolyticus are the leading cause of seafood-associated illnesses that causes about 80,000 illnesses and 100 deaths a year. Infection can occur through eating raw seafood or through an open wound. The studies have shown that Vibrio has a high frequency of antibiotic resistance in the environment. Next. Okay, so in this picture depicts in 2010, the largest oyster ground was shut down, and it hasn't been shut down in over a decade due to Vibrio vulnificus infecting 38 people. In Louisiana, the oyster industry accounts for $317 million a year and provides over 4,000 jobs. 70% of the oysters harvested in the US originate from the Gulf, making this industry important for Louisiana, but also for the country as well. This study in 2010 by Han et al. Attempted to show how susceptible the Vibrio species were found in oysters collected in the Gulf of Louisiana, as well as at retail stores. To see how they would be affected by various antibiotics. They found that 2 Vibrio species were susceptible to the antibiotics, and another major result was that Vibrio parahemolyticus was not, was not susceptible to an antibiotic called ampicillin, which can potentially pose a human health risk. Next. So this is fairly important to not only the Gulf, but anywhere that Vibrio is found, and it is found nationwide, as well as PPCPs are found worldwide in the environment. Reported concentrations are fairly low, but with an increase in population size, this can potentially change. MAR stands for multiple antibiotic resistance, which has been detected in Vibrio, and this means the Vibrio species are resistant to multiple antibiotics. Animals have also been shown to have MAR such as shrimps, dolphins, and seals. Can you click? The role of PPCPs in the environment has been focused only on a few types of antibiotics in the literature, and there is a need for more research to better understand how these PPCPs are causing the selective effect for antibiotics, as well as how this will affect human and animal pathogen, and thus also affecting human health. Next. So to end on a positive note, there are some suggested solutions within the literature, such as enforcing the methods that further reduce and remove PPCPs from the wastewater before they are discharged directly into the environment. Another potential solution would be creating medicines with various physio and chemical structures so that they better biodegrade in the environment. As well as creating a better drug delivery system and disposal available to the public, as well as companies that produce these PPCPs. Although more research is needed on management and mitigation options to make a better informed decision on the best way for dealing with this emergent pollutant moving forward. Thank you for your time, and I will take any questions.
Thank you. Do we have any questions? I see lots of nope and no popping up.
Thank you.
Okay. Thank you very much. I guess we will move on then.
Okay, can you hear me?
Yes, we can.
Okay, my name is Tasha, and I'm going to talk to you about bioremediation techniques. So you can flip to slide 1. Can you click? Yeah. So I'm going to briefly go over what bioremediation is and different types of techniques with some pros and cons. Just lightly touch on effectiveness and limitations, and then go into some potential examples of how it could be used here and looking forward. You can click. So what kind of problems can bioremediation solve? There's all different types of pollution. So there's chemical plant wastes like PCAs and PCBs, gasoline and petroleum products, dyes from textile factories, radioactive waste, heavy metals, including arsenic, cadmium, chromium, mercury, nickel, cesium, and a multitude of pesticides. To like runoff from hog and cattle farms, fertilizer from crops, oil spills, and leaks. You can click. So to define bioremediation, I would say that it's the use of naturally occurring organisms like bacteria, plants, and fungi to recycle harmful organic contaminants into benign compounds. So it removes pollutants, fertilizers, heavy metals, and other toxins to restore the ecosystem to a healthier state for its human, floral, and other faunal inhabitants. In most cases, the main limiting factor is the presence of oxygen, followed a little further down by nitrogen and phosphorus. So there are 2 different types. Generalized types are in situ and ex situ. In situ meaning in place, like where the actual contaminants are, and ex situ meaning that you remove the contaminated source and transport it elsewhere, treat it, and return it. So there's different strategies for soil, groundwater, and open water. For soil, in situ has aerobic and anaerobic types. And for aerobic, there's bioventing. So they pump air and oxygen, and sometimes with the addition of nitrogen and phosphorus to oxygenate the soil, or they inject hydrogen peroxide into the soil. And they only do that in areas that already have contaminated groundwater, and it encourages the natural bioremediation process that is carried out by bacteria in the soil that's already there. All of it requires knowledge of what kind of contaminant you have in the soil and what kind of microbes are there that could be utilized to remediate the soil. For anaerobic, it happens naturally. Methanogens use electron receptors to metabolize contaminants, and if they add sulfates or nitrates, it can enhance the process a little bit and speed it up, but it's not as fast as aerobic. For groundwater, there is something called air sparging and soil vapor extraction, where they force air into the groundwater and it reassigns the contaminants to vapor form, and then they vacuum it out of the soil. So for ex situ bioremediation, soil has slurry phase and solid phase forms. Slurry phase is where they remove contaminated soil, mix it with water and additives to instigate bacterial remediation, and then the water is removed and the soil is replaced. Solid phase, like land farming, biopiles, and composting, remove the contaminated soil and treat it by aeration and sometimes with nutrients until it's clean. Some of these are a little slower than the air sparging or the aerobic bioventing. And they're more expensive as well because you have to remove everything and bring it to a different location to treat it and then return it. Groundwater can be removed, reconditioned, and replaced through injection wells. Can you click? So to hone in on the three main things that we're looking at for bioremediation, microorganisms is the first one. Pseudomonas butida KT2440 remediates toluene from paint thinner and naphthalene from petroleum refining processes. It's genetically engineered. So we made this, or a version of it, that is very effective. The next example I have is Deinococcus radiodurans. It's also a genetically engineered microbe, and it is radiation resistant, and it's an extremophile. So it metabolizes mercury and toluene. The third one is Alcanivorax borcumensis, and they used this in the Deepwater Horizon oil spill to remove hydrocarbons, and it was a major player in degrading the oil from the spill in the Gulf of Mexico because it removes aromatic hydrocarbons and breaks them down into benign elements. Can you click? Phytoremediation is the use of plants for bioremediation to remove these harmful chemicals. So the first one you guys are familiar with is Spartina grass, and it is used to absorb the number 2 fuel oil, like the Louisiana crude in the marsh. So the roots are what absorb it out of the soil. So being a native plant, that would be default, like what we're looking for, because we're not introducing a foreign organism. The second one is a microalgae called Chlorella vulgaris, and it reduces nanoparticles of nickel oxygen to a 0 valence form, which is less toxic to the environment. The third example I have for you is Salix nigra, which is the black willow tree. It's common to riverbanks and wet areas, and uptakes bentazone, which is an agricultural herbicide, into its leaves, where when the light hits the leaves, it helps the plant degrade the toxin. Can you click? The last way for bioremediation that I'm going to talk about is mycoremediation, using fungi to clean up the environment. Pleurotus austriatus mushrooms, which are oyster mushrooms, are tolerant to salt water, and you can use booms and made with hay or straw, and you infuse them with the mycelial mats, and they grow, and they can grow and float on the water and create a physical barrier, but at the same time absorb it and break down the oil into non-toxic forms. Shaggy mane mushrooms, which is Coprinus comatus, and phoenix mushrooms, Pleurotus pulmonaris, remediate heavy metals. They uptake and concentrate heavy metals into their fruiting bodies, which can actually be harvested and brought to recycling plants, cooked down, and harvested from the actual mushrooms. Shiitake mushrooms, which you're all familiar with, Lentinula elodes, remediate polycyclic aromatic hydrocarbons that result from burning coal or gas, and also PCBs, which come from coolants and used oil, and PCPs from pesticide and treated timbers. So mushrooms are, if you use naturally occurring ones that are local to the region, they're pretty effective at cleaning up the land. So it's another thing that isn't widely used because it's kind of a newer technology, but it's something that could work together with the others to help. Can you click? So effectiveness and limitations. In situ, whether we're looking, regardless of what type we're looking at, it is faster generally and less expensive than ex situ. And it generally requires oxygenation for microbial, and you need to know, you need to do some testing first to know what the makeup of the soil is and what the makeup of your contaminants are, how much your contamination is, because a lot of times if there's too heavy of a saturation of contaminants, it's going to overcome the bacteria or the plants or even the mushrooms, of course. So it's definitely something that all of these can be used, but they may need to be used in conjunction with mechanical or chemical technique as well to aid them so that the organisms aren't overwhelmed by the contaminants. A lot of them can also be sped up by applying inorganic fertilizer, and it's good because you can use local organisms, so it's less impact on the environment. For XC2, it's easier to control the variables that have effectiveness. So especially like the solid phase strategies are kind of slow. Slurry phase works well and more quickly, but they're a lot more expensive and it requires a lot more work and intrusiveness to the environment around them because you're physically having to remove everything and then return it back later. You can click. So for solutions, obviously all of these different technologies, whether new or well-established, are capable in their applications, but every event is going to be different. So you would need to evaluate the event first before deciding what types of strategies need to be used. And looking forward, I would think that maybe something preventative would be a way to lessen the impacts of these events when they happen. If you already have these things in place, you can plant these plants on the riverbanks. You can already have mushroom farms growing and bacteria farms growing so that when it's time, you can just apply and already have it ready to roll, and it would speed up the remediation processes. So that's pretty much all I have for you guys. I know I talk really fast. So are there any questions?
I have a question.
Okay.
When you on the Spartina graph, when it uptakes, when my presentation's on Thursday, when I did my research, I had found that when oil is introduced to the Spartina alterniflora, they actually exhibited high mortality rates. Is the high mortality rate, and is that due to the uptake of the specific hydrocarbons? Or do you know?
That's what I understood from what I read, that if it's overwhelmed, it will still bioremediate, but it does affect the reproduction of the plant like down the road. So it'll do the job, but it may inhibit the plant from reproduction.
All right. I was just making sure that I was reading my research right too. Thanks, Tasha.
Mm-hmm. Sure.
So just to clarify, obviously, in terms of bioremediation of oil, most of our wetland that was oil was Spartina alterniflora, and much of it died and was lost.
Right.
So, you know, the different plant vegetation types have very different degrees of tolerance to oil.
Right.
So as you see shifts in vegetation, some areas that are dominated by one species as opposed to another are likely to see different types of responses to being oiled.
Right, and especially with the magnitude of the oil.
Yeah, alterniflora is a bit more resistant than some of the other dominant species like Spartina patens or Disticulus or Juncus, but it is still quite sensitive to oiling itself.
Thank you, Dr. Robert and Tasha.
Okay. Any other questions for Tasha?
Okay, so if those plants are susceptible and they die, how well are they going to? If you go back and replant those, are they going to be able to grow in those areas after, like, say, a few months or a few years, or how long does it take for the soil to recover?
The soil itself, for natural processes, because of the nature of marsh soil, it's a lot of anaerobic it's going to take a longer time without any extra help from bacteria for aerobic on the surface. But as you get deep penetration of the oil into it is, that determines how long it's going to take for it to be naturally bioremediated. So it could be that if you plant some, that it would be able to grow. It just kind of depends on how much contamination you have and what the nature of the specific contaminants are as to how fast they break down even naturally before they're at a level that isn't toxic to the Spartina. Does that make sense?
Yes, thank you.
Okay, we don't really have time for me to go into a long answer to that question, but you have to realize that the oil is degrading over time itself through the microbial community that's there, and we had quite a bit of success with replanting Spartina in oiled sites. So, but there is a lot of variability that exists in those. Okay, thank you very much, and let's move on to the final presentation today.
Can you hear me?
Yes, sir.
We can.
Okay. Let's see. You'll let me have time to do this. So you can go to slide 1. My name's Austin, and I'll be talking to you all about coral bleaching. So coral bleaching is the result of the algae species leaving their coral host in the surrounding environment because of rising temperature or pH. The temperature doesn't even have to drastically change by that much, even a degree or two can just implement these changes. And so the resulting problem leads to the interesting question of how these corals and algal species adapt and change, if they can, to the changing conditions. The focus on their symbiotic relationship between them is just as important as focusing on the rising sea temperatures that affect them. Next, please. So corals are an invertebrate species in the class of Anthozoa in the phylum Cnidaria. They're colonial polyps that form together, and they're sessile individuals that can catch and consume zooplankton and small fish with their nematocysts, similar to other cnidarians like jellyfish. So, however, most of their nutrition comes from the symbiotic relationship with the dinoflagellates, and they form the reefs by forming their outer skeleton using calcium carbonate that they sequester throughout their lives. And as the corals die and other invertebrates die around them, like mussels and clams, the calcium carbonate skeletons build up the reef and increase the size of it. And because the algal symbionts need sunlight, especially to produce nutrients, most corals are found in shallower waters, although some can live in deeper depths. And the symbiotic algae relay their carbohydrates they sequester to the coral body for nutrition, while the coral provides shelter and nutrients as well. Next. So you can think of the relationship in terms of a basic dynamic energy budget, where each using disparate nutrient building processes that complement one another. This relationship is a beneficial mutualism. However, it did start off like all mutualisms as exploitative millions of years ago. The Symbiodinium species live in the oral endoderm, where they undergo their own metabolic processes while simultaneously exchanging their metabolic products with the anthozoan host and vice versa. Next. So this is a basic diagram showing the Symbiodinium clades, the algae on the left, and on the right are the different hosts that can have them and host them. So some Symbiodiniums display a wide range of host specificity, which are more generalist symbionts, while other species are highly specialized in the symbiosis with certain invertebrates. So an example of high specificity are displayed by certain foraminiferous species, which are single-shelled protists that only form symbiosis with the members of clade F. and the giant sea anemone that only form relationships with clades A and B. So some hosts display different ecological and environmental conditions for symbiont specificity, an example being Zoanthus sansabarku, which associate with different symbiodinium based on the depth that the Zoanthus is at. So it can favor either a more sun-adapted or shade-adapted algae that have a symbiosis with. So this ability to shuffle around different algal species as a host is beneficial, especially in a locally changing environment with different light intensities, turbidity, and temperature. Having functionally diverse strains of symbiodinium floating around, each of which has their own genetic diversity, along with host flexibility, allows a host to respond to different consistently variable conditions in its environment, also leading to the constantly changing microenvironment for the algae within the host itself. Next. So main problems obviously facing coral reefs are rising temperatures and pH in the oceans. And the main problem with this is that corals in general have, and their symbiosis with algae, have a long evolutionary history. So the main concern is that the accelerating rate of environmental change could exceed the evolutionary capacity of coral and their algal species to adapt. A common view is that corals are too long-lived to evolve quickly, and the geographic differences in temperature tolerances have evolved over much longer time frames than the decadal scale of current changes in climate now. So although some corals are indeed very long-lived, sexual maturity is reached in about 3 to 5 years, and most species at all depths rarely live longer than 20 years. Nonetheless, highly skewed fecundity distributions, where a very few large, old individuals swamp the gene pool, with strongly overlapping generations and high levels of asexual reproduction, are common traits that are likely to retard the rapid evolution in many coral species. So the evidence right now indicates that rates of gene flow in corals varies substantially among species, which implies that their differential ability to migrate in response to climate change and to adapt will result in further changes in community structure beyond the immediate effects of selective mortality caused by source bleaching, severe bleaching. In contrast, subpopulations on isolated islands or archipelagos, for example, Hawaii or Bermuda, may represent genetic outposts for virtually all coral species. So, for example, if an island reef is bleached, their recovery time might be much slower, or more than or faster, depending. And obviously, different locales have varying changes in the temperature and other conditions, and so species that live there may have disparate adaptations for where they live and evolve. And so some species will be able to respond more rapidly than others, resulting in community structure of these reefs to change vastly over time. Next. So some species of corals, algae, and other invertebrates have already started responding to these changes in climate. The changes in community structure of today have already led to other species responding, but bleaching in areas, especially large areas, is very patchy, which basically shows that some species have higher tolerances and adaptations for responding to the environmental changes. So basically, changes to the Great Barrier Reef and other reefs from bleaching and other human influences have consequently had species who can more rapidly recover, reacting to these changes, with taking over areas where other species fail to respond to these changes, basically just changing the community, and which would also affect top-down and bottom-up processes and trophic levels. So this will have interesting effects as the years go on. You can go next slide, please. So there's a little bit of hope here with, or a possible hope with, a certain clade in the symbiodiniums, clade D. Many species, or most species in that clade, have been shown to be much more resilient to warmer temperatures and bleaching events than any other clade. A study by Stats and Gates have identified clade D symbiodinium as uncommon, pandemically distributed generalist, found in higher abundances on reefs that have been exposed to changing sea surface temperatures and local stressors, or with a history of bleaching in general. So they're basically show better responses to bleaching events than other clades. And this distribution suggests that clade D are most likely opportunistic endosymbionts, whereby they outcompete and replace the optimal symbionts in a health-compromised corals. So this is a good thing. However, there are obvious drawbacks with any ecological change. And the reason that clade D symbionts aren't already taking over local and global reefs is the fact that clade D species are a much more slow grower than other species. And while they accumulate on hosts that were damaged from bleaching, they also leave these hosts more susceptible to opportunistic symbiodiniums that may just use the corals as strictly habitat rather than a functional mutualistic relationship, which would switch back to the exploitation relationship rather than a mutualism that benefits both. So it has good and bad, advantages and disadvantages. So hopefully in the future there will be more studies geared toward clade D and the species. I believe it's clade either C or G that also displays some form of have some species that have some temperature resilience as well, but the majority of these species are in clade D. So you can go to the next slide. So, future directions, manage obviously climate change and CO2 emissions, implementing policies that protect areas where more vulnerability to change is expected, and obviously more studies geared toward clade D and other species that would buffer coral reefs from warming oceans, and having studies on overall ecological changes in these areas, because even though clade D might help buffer short time changes, long changes to the trophic levels from bottom up processes and top down would be affected as well. So studies should be geared toward that as well. So that's it. Any questions?
We have one question from one of your classmates. Did you see how the disappearance of coral reefs would affect fish life?
I haven't really looked at that too much. I haven't seen too many studies involving that yet. I know that there is one coral reef somewhere I saw where obviously changes have been implemented, and I believe a few of the locally adapted fish species, some responded while some died off. So obviously the top down effects and bottom up effects would change the overall species that live in that area. I haven't seen it that much yet, and I think most people haven't fully looked at that as of now. I think that'll be really a question that's answered probably in the next 5 years, 10 years maybe. So.
Other questions for Austin?
Yes, there is another question. So does the bleaching of the coral only, sorry, been a long day. So does the bleaching of coral only signify the organisms leaving the host, meaning that the coral host itself is still alive?
Yes, however, the coral host can still survive for a very, very short time. This is basically called the recovery time, and during this time, either a coral takes on other algal symbionts, which really is where clade D comes in. Clade D would basically just be floating around in general, and those species, as soon as they can attach to a host that has been bleached or needs new algae species, they would attach to them and hopefully help them recover. However, if they don't, I don't know the exact time frame because it's different for different coral species. But if a coral doesn't receive new algae species, then the host and the coral in general will die permanently. Yes.
Okay. Well, I want to thank all of you for giving the presentations today. It's a really nice start to the final week of the class. And we have 4 more presentations that will be coming up on Thursday. And if you have any questions in the meantime, let us know. Otherwise, we will see you Thursday at 11. Thank you.
Hey, Dr. Roberts, I have a question.
Yeah.
I just have to have my presentation slides to y'all by midnight the night before.
I'm sorry. I'm having a hard time hearing you with the volume. Can you just speak a little bit louder?
Yeah. I just have to have my presentation slides given to y'all by midnight the night before.
Ideally, you would have it in the afternoon the day before, so that way we can convert them to the slides, so that way the rest of the class can have them available to them.
That's fine. I can have that. That's fine. They're already done.
Okay.
All right. Thank you so much.
All right. Thank you.
Bye-bye.
Bye-bye.